So good morning, everyone. How's been your day and how's been your night? This is another opportunity for us to learn one again. This is the voice of Mr. Adil Latunde, and you will recall that we actually started the first part of this presentation, this lecture, and that's why it is titled "Still at the beginners level in microbiology." This is microbial evolution and ecology. In the last slides, specifically explain what microorganism will actually mean. And by the assignment I gave you, which I gave you that you should go and draw a typical all the classes of microorganism. You should be able to know how do they actually look like and what are the components of each microorganism. Okay, so and I told you that when we talk about evolution, I try to explain what evolution is all about. That is development with modernization, or with what we can call advancement. And I told you that sometimes ago you find some microorganism in a different habitat, in a different environment, but in an attempt, according to the theories of evolution, one of the theories of evolution was that of Charles Darwin, which actually talked about the survival of the fittest. That is, if you found an organism in an environment, organism, it could be microorganism, it could be any organism, so to say, if you pick it up now and you want to adapt it to a new environment, such organism is going to develop some attributes that will make them to be able to adapt to that environment. Then, apart from the theory of Charles Darwin, another theory of evolution is about the theory of use and disuse. And what is this theory of use and disuse? That is any attributes or any features or any characteristics that you are using continually or continuously, you are going to get adapted, you are going to improve on it. And that is what is called evolution as well. And I told you what ecology is also all about. That is ecology, it is ecology that has to do with the study of the interrelationship between the ecosystem or between the environment in where you found an organism and that organism in particular. And the summary of what we are trying to say is just that microorganisms, wherever they find themselves, they have to establish a kind of interrelationship. That is their new environment. So, and that's why we actually explain that of microorganism in the guts of a human being. You will know that the interrelationship could be in form of parasitism. It could be in form of symbiotic relationship, which has to do with the mutualistic form of existence, and so on and so to say. So you agree with me about that. Then again, I also told you that in this course, the origin of anything or the beginning of anything is very, very important. And that is why that when you pick up microorganisms today and you try to study them with respect to the way they interrelate with their host, that is a human, or it could be woman, it could be so with their host. We just discovered that two of them, both the host and microorganism, they have developed over, they have developed over certain times. That is, they've developed certain kind of. So the more, just like they say in Yoruba that tewe bakolara ose, o ma dose. That is, what we are saying in essence is the more intimate you become with your host, the more intimacy about your relationship with your host, the better for both of you. Okay, and that's why we cannot even do without the origin of life. That's how did the microorganism actually start their life? How did they look like? What was the structure all about? So, and that was what led us to talk about prokaryotic organism as well as eukaryotic cell. I told you then that prokaryotic organism, pro from the word primitive, from the word before, from the word, so that is um, that is from the word primitive. And I told you that carriers or from carrion, which has to do with chromosome or nucleus. So one of the distinction between two of them is that in prokaryotic organism you have pseudo, you have primitive nuclear material. So and apart from that, that they are not they don't have organelle. That is, they are not membrane bounded. They don't have any distinct organelle. There's no clear place whereby you can say this is the nucleus. Unlike what you see in eukaryotic, whereby you see that this is their nucleus, and we believe that the nucleus is where the DNA should actually reside. So, unlike that, we talk about all this, and we were able to know the differences between prokaryotic organism and eukaryotic organism. Having said that, we also just like we also know that in this kind of presentation, there is all necessity that you should understand microbiology beyond just the science of studying microorganisms. You should understand it even in their chemical composition as well. That is, it has to be in their alphabet. So, and that's why we said one of the learning outcome is to be able to apply chemical principles to be able to tell us how microbial function is going to look like. For example, when you talk about a fermentation, very simple fermentation, you just see that a fermentation is a metabolic process whereby there are kind of interrelationship between microorganisms, well, sorry, interrelationship with the respect to production of certain chemicals. For example, now when you talk about glycolysis, it has to do with the breaking down of glucose into pyruvic acid. So how is glucose going to be broken down? That means it is going to take a lot of processes, that is enzymatic processes. So if you do not know how glucose is looking like, you might not be able to understand microorganisms in that way. And that's the reason why I also mentioned that this course, you should be able to know about the chemical principles of microbial function. Then you should be able to compare, just like I said, prokaryotic cell and eukaryotic cell. And you should be able to know their anatomical components and their function. So then, apart from that, you should be able to describe certain simple metabolism, and I define metabolism for you as the process, certain processes which can be divided into anabolism and catabolism. When you talk about anabolism, that is breaking down, and when you are talking of catabolism, that is building up. Two of them will start with B, building and baking. So, and we'll be able to give certain types of, you'll be able to pick some processes, some physiological processes like growth, like nutrition, like photosynthesis, like respiration. You'll be able to pick them and be able to identify whether this one is going to be catabolic, this one is going to be anabolic. So as well, then you'll be able to understand them in form of their growth, because the more they have access to nutrients, the more they will perform. Just like a human being as well, the more well you feed, uh, your function, the function of your existence will depend on the quality of the food you are taking. and that is if you are not feeding well, it's going to affect your immunity level of immunity. You're able to resist uh, diseases and so on and so forth like that. So we wouldn't have time to go into that. Then I also said I made mention of the principle of what is called recombinant DNA technology. Another word for recombinant DNA technology is what is called genetic engineering. That is, you microorganism is such a way that you can use microorganisms to be able to produce some enzymes that are very, very necessary or which are deficient in human. For example, we have been able to be successful in recombinant DNA production of certain enzymes, like the enzyme of diabetics, okay? So, and so on and so forth, like that. So, we know that it is very relevant in health, environment, food, and industry, and so on and so forth in our hospital, in our hygiene, and so on. Then we'll also talk about key definitions like microbe. What do they mean? Microbiome. I told you, collection of microorganisms that live in the same place and their gene in a certain habitat, and pathogen that disease causing organism in a specific hole. And just like I said, symbiosis, that is a close and but long-term biological interaction between different organisms. We also look at the association that can be in form of commensal or commensalism, which leaves commensal animal lives with a host with no obvious benefit or harm and opportunity. They take that opportunity of naturally they are not there to cause any harm or disease, but as a result of maybe immunocompromised situation, you will find them now causing infection. And we talk about a certain milestone and history in microbiology. So we'll look at that of Hooke and Leeuwenhoek, Pasteur, and Koch postulate as well. Then I also made mention of what do we mean by microbial diversity, that is bacteria and archaea and eukaryotic microorganism acellular, acellular, that is absence of a certain cell, that's the group that contains viruses, viroids, prions, and so on. Now, let me, at this point in time, let me just inform you or tell you and even educate you that, let me say, nowadays, there is something we call the WOESE. If you want to classify microorganisms, we call it WOESE classification. WOESE classification was based on ribosomal RNA. That is a rRNA. So that classification was more of molecular classification. It was based on the fact that RNA will not change with time, will not undergo mutation with time. DNA can undergo changes within a very short period of time, but over millions of years, RNA will not do that. And the RNA now happen to be the signature. It happened to be the signature for classification of these microorganisms. So, and WASIC now classify all living organisms. We classify them into three categories. The first group, we are going to see it in subsequent lectures. The first group is called the group of archaea, which are the most primitive, more primitive than even bacteria. So archaea. Then the second classification was on prokaryotic, that is the prokarya, archaea, prokarya, and the third one is eukarya. Eukarya. We are going to see more of this about WASE classification, and that is the one we are still making use of it up to today. And I told you that if you synchronize all biological agents, including man, if you synchronize them, you will just discover that they are made up of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So, which occur in very dominant way. And you should be familiar with what are the bond, that is interaction between certain molecules. So, for example, ionic bonding. For example, when you pick up the DNA, although DNA is a biological agent, but when you pick them up, you just discover that there's going to be part that's going to be the sugar backbone. There will be an aspect that's going to be for phosphorus. There will be an aspect that's going to be for oxygen, and so on like that. So, if we do not understand how bonds behave in all these chemicals, you might have it very, very difficult to be able to understand the prospect. I mean, the simple prospect, even prospect of microorganisms. You won't be able to know what they can do, what they are capable of doing. You won't be able to even understand their structure. So all these are very, very important. And you should know that water is very, very special. And that's why they call it a universal solvent. It is present in virtually everything, all living organisms. In fact, there can never be any life without water. If our blood is made up of 90% water, so who are we that will not say that water is not going to be very, very important? I made mention about pH buffers and macromolecules. So pH, that is the study of alkalinity and acidity of a given microorganism. And each microorganism, they have their optimal pH, that is the pH at which they can perform best, at which they can exist without any problem. Just like human being, once you have all the basic needs of life, basic need of life, you have shelter, you have this one, you have that. So at that point in time, you just discover that you are performing excellently. So the same thing with a microorganism, once they are in existing in their normal pH, optimal pH, you just see them, they won't misbehave, they will just be doing very much, they will be doing very well. So, and don't forget that buffer resist pH change. It is because of buffer, that one will not allow the pH to just change, just like for example just move from 3 to 10. No. So because there are certain solvents that are also very present in microorganism that will also make sure that certain changes are highly restricted. And micromolecules like carbohydrates, lipids, proteins, and nucleic acid. So all these four are basically what is very, very important in life. For example, carbohydrates, if you don't have any carbohydrates, you won't be able to carry out cellular activities like cellular respiration, photosynthesis, and so on, because carbohydrates glucose is carbohydrate, and the breaking down of this one is going to give us CO2 and water, so that the organism is going to is going to respire very well. Then lipids as well, we know that lipids are very, very important, especially when it comes to the issue of insulation. And by the time we get to the structure of the cell wall, you will see that the presence of lipid in some microorganisms has actually helped in their classification as well. And sometimes lipids are also source of energy. That is, when carbohydrate is not in plentiful supply, the cell can decide to convert lipid into usable form, so that glycolysis or any other physiological process will be able to proceed without any problem. And you know, protein, when you talk about protein, form of enzyme, that there's no catabolic or anabolic processes or biological processes that will ever take place without enzyme. So that is very, very important. Then apart from that, we also talk about nucleic acid like RNA, DNA, and so on and so forth like that. Okay. So enzyme, we have actually mentioned this. So then you also mentioned all this about gram positive, gram negative, and waxy or mycolic acid, which actually classify them into acid fast. I will want to just rush through this so that you're able to do. So all these internal structures or internal features, they are very, very important, so that you're able to know and how this is a structure of a eukaryotic cell. And you can see that this one has contained membrane-bounded organelles, just like you can see Golgi apparatus, mitochondria, nucleus, and so on and so forth like that. And the cell wall and the membrane. We also look at comparison, comparing them together, just like this. Now, this is where we stopped the other time, which we talk about metabolism, which is the big picture. The big picture is metabolism, what you see, the activity the main activities of the body. I mean, in the body is metabolism. It is either the reaction, biological reaction, it could be in form of catabolism, which is breaking down, or it can be in form of anabolic, which is breaking down. Okay, and just like I building up, and just like what I said, that ATP is known as the currency of the cell. Every cell, that is the ATP, is known as the naira of the cell. So, and we also have some, not only ATP, we also have some redox carrier, that is some currency as well, but they are not in form of ATP, but they are in form of coenzymes. So we will see all this as we go on. Then, just like I said, that you should know glycolysis is the breaking down. You will learn this in microbial biochemistry. So it is the breaking down of glucose into two molecules of pyruvic acid. Now, when you look at glucose, glucose is C6H12O6, that is 6 carbon atom. So when you now break the 6 carbon atom, the first of all is that it's going to be broken down into fructose, fructose, then at a point in time it's going to be divided into 3 carbon compounds. So three, this phosphoglyceride, aldehyde, and so on. Then with the eventual or the end product to be 2 carbon of 2 molecules of pyruvic acid. Pyruvate or pyruvic acid, this compound is a 3 carbon compound. Glucose is a 6 carbon compound. So when you now when you divide 6 into 2, it's going to be 3. That's why the molecule is going to be in 2. So glucose or glycolysis is the breaking down of glucose into enzymatically breaking down of glucose into 2 compounds or 2 molecules of pyruvate, which is a 3 carbon compound, then plus small ATP, which I call the energy currency, and the other electron carrier, which is a NADH. So you can see that is what you can say generally about glycolysis. So, and just like I said the other time, and I will continue to say it, that nowadays you can now have the opportunity of simulating using on your phone or your laptop. You'll be able to see how glycolysis is being broken down into all this one, so that it's going to come live to you. And you should know that glycolysis occurs in cytoplasm. This should be established for you. It occurs in cytoplasm, not inside the nucleus. It takes place outside the nucleus. Don't forget that cell can be broken down. You can divide cell into 2 compartments. The first compartment is cytoplasm. The second compartment is nucleoplasm. And when you come across the word cyto means cellular or cell. And when you are talking about plasm is the liquid content or the liquid part of the cell. Don't forget that I told you how important water is to all cellular activities, that there's no cellular activity that can proceed without water. So cyto means cell, plasm means water. So the liquid parts. So if they say that you have blood, the blood of every cell, the blood of the cell is cytoplasm. Yes. So glycolysis occurs in cytoplasm and in the absence of oxygen. So it is an anaerobic process. It is an anaerobic process where glucose can be broken down or is broken down into 2 molecules of pyruvic acid. So because no oxygen is required. Now, the end product, which is a pyruvate, if you understand if you understand glycolysis very, very well, you will now discover that this pyruvate acid, in the presence of some enzyme, is going to proceed to fermentation. That is the immediately you have, don't forget that I said pyruvate acid is three carbon compound. And from the three carbon compound, in the presence of some enzyme, three will now move to two carbon. The two carbon we now talk about is C2H5OH. That is a ethanol. That is ethanol, which is the product of fermentation. So what you are saying in essence is the end journey, when you pick up glucose, the cell will make sure that when you talk about okay, we breathe in oxygen, we breathe in oxygen and we send out carbon dioxide. Okay, that carbon dioxide is a single carbon compound. And if it is a single carbon compound, the overall goal of you taking up glucose or taking up carbohydrates is that your ability to be able to break down that glucose, which is six carbon compound, your ability to utilize it and be able to produce just one carbon compound, which is CO2. Now, we have gotten a process where the glucose can be broken down into three carbon compounds. Yes, three carbon compound, which is called pyruvate acid. So the journey will not stop there. From glucose to this is called glycolysis. And from the this end product, this is it's just like somebody who is preparing or want to get to Lagos from Shaki. And when you get to Isheyin, that is just just a stopping point. No, sorry, that's not the end point. Don't forget, you are going to come across a lot of many towns. You come across many towns, villages before you're able to get to Lagos. So the journey from Shaki, what you have is what is glucose, which is a six carbon compound. And when you get to Lagos, your Lagos should be, which is the destination, should be that you should be able to have gotten a process that will make you to be able to move from Shaki, that is glucose, and what you are going to have is going to be CO2, which is carbon dioxide. Okay, so when you now get to Isheyin, you have not gotten to the end product, to the end or the destination. So that is two pyruvates. So, and that one will involve no oxygen and it will take place in the cytoplasm. Now, the first destination from Isheyin should now be some of this term, and the next term is going to convert the three carbon into two carbon, which is going to be C2H5OH. So by the time you understand, we just go and pick up your glycolysis in microbial biochemistry and study it very well. You just discover that glucose, there are 10 steps from glucose to pyruvate. There are 10 steps. So from that 10 steps, if you are going to, the journey does not stop there. There is going to be one point whereby the C3 carbon is going to be converted into malic acid and all this one. That's going to be acetyl CoA. That's going to be that's going to proceed to what is called, or where it is called um Krebs cycle. So fermentation, that is a production of C2H5OH, that is ethanol. Okay, now TCA is very, very important. TCA, that is a tricarboxylic acid cycle. Tricarboxylic, that is three carbon compound, which is pyruvate. I just mentioned it now. Pyruvate is going to move from pyruvate to acetyl CoA. is acetyl coenzyme A to TCA, and this is going to be captured in electron. It's going to be in form of this. So by the time you now read, make sure you read. This one is Krebs cycle. It's Krebs cycle. So you are reading to know the difference between aerobic and anaerobic. That is, you want to know the difference between glycolysis or fermentation, as well as the difference between and when you compare it with the aerobic process, which is a tricarboxylic acid, or what you can call the Krebs cycle. Okay. Now, just like I said, you should be able to differentiate between fermentation and anaerobic respiration. Okay. Now the difference is that fermentation regenerates NADH, and the end product is going to be in form of lactates, ethanol, and other acids. So you should let that one be established with you, that the end product of a fermentation is a lactate, ethanol, and other acids. So, and that is the reason why that, and there are some microorganisms that their own job, what they need in their own life, what they will consider as the success of their own life is just their own ability to be able to metabolize glucose into pyruvic acid or pyruvates. Whereas the journey of some will actually start from where their own has ended and is going to proceed via fermentation, especially microorganisms that are involved in fermentation process. um So their own, even if they have 10 molecule of pyruvic acid, they are not so happy. They have not gotten to their own destination. So their own destination is a place whereby they can have lactate, ethanol, and other acid. That is when their own life is going to start from. And anaerobic process, which is um, for example, which we can call anaerobic process in industry, like glycolysis, uses non-oxygen acceptors. They depend on non-oxygen acceptors. So this can be applied in food, in diagnosis, as well as ecology. So we will know that. Then what do we mean by phototrophy and chemolithotrophy? So when you come across the word photo actually means light. Eh? That is the ability or some microorganism that their own ability is they metabolize or they convert light into chemical energy. And that chemical energy, example of such bacteria or example of such microorganism are those one we call cyanobacteria. Cyanobacteria. Okay. So they are called phototrophs because they metabolize or they convert light into chemical energy. Then we also have some that are called chemolithotrophs. This one oxidize inorganic compound like ammonia, um hydrogen sulfide, and iron 2. So they oxidize it. They have the ability to oxidize it to the form in which they need them. So for example, all this one is what we have in the nitrifying bacteria. All these nitrifying bacteria that they can be used as fertilizer and all this one. So they are called chemolithotrophs. So there are still going to be um, detailed discussion about this. So environmental importance of chemoliths is that primary production and nutrient cycle. Some of these nutrient cycle like a nitrogen cycle, carbon cycle as well. All these are what we actually need. All these microorganism form. Then again, now let's now consider what is called the microbial growth. Microbial growth. What do we mean by growth? So when we are talking about growth, that is irreversible, irreversible, huh? In size. irreversible. That is, in bacteria or in microorganisms, there are two different. We have binary fission. That is, when you have one divided, and which is going to be the generation time. So when you are talking of microbial growth, these are very, very important. You should be able to master the growth curve. The growth curve is going to start from lag phase, which is a non-dividing stage. This is a stage whereby the microorganism is just getting used to the environment, just getting used. For example, when you pick microorganism from one um, habitat and you now culture it, you introduce it into another. So the growth will not just start immediately. They have to accumulate some certain chemicals. They have to accumulate, they have to get accustomed. So that is why it's just like human being. When you pick somebody, no matter how lousy the person is in a new environment, they are going to take some time for them to be able to master their new environment. The same thing with microorganisms when they are talking about the growth. So when you introduce them, so at large phase, they are just not defined, they are just mastering, they are getting accustomed to um, the nutrients and the environment. So after getting accustomed to it, so they will now proceed into the log phase. That is now the defining stage, log phase. So they will develop until they now reach a stage whereby they must have utilized, they must have finished or almost finishing or getting to the end of it. So it's just like somebody that you now give them food. You now say, "Oh, before I come back, I am traveling. Before I come back, this is a bag of rice, a bag of this, a bag of that, a bag of that." So, and before they will know it, so that means they will get accustomed to it, and by the time they get accustomed to it, they will start eating. By the time they are eating it, they are developing, they are happy, they are this. So until when it now comes to them, now say, this thing is getting finished. So when they now get finished, they will now talk to them and say, please let us reduce the intake so that it is going to last them until when their father must have come back. So, and that is at that stationary stage. So they will still be eating, but there won't be appreciable growth. So that's why it is this one is going to be exponential stage. Is going to increase rapidly. The growth curve is going to increase rapidly. So until it gets to a point whereby it's just going to be what? To be flat. That is at stationary stage. So and at any point in time, when they must have utilized all all the component, I mean the nutrients in that. So the population will now begin to reduce. So they will have now entered into the stage of death. Okay. So now you should one of the reasons why you must know and master the growth curve is that you will be able to use it for your culturing in the laboratory, for your isolation in the laboratory. So, and by the time you are using it for that, it's also very, very applicable in drug production. Now, growth requirement and oxygen classes. So, some bacteria can be classified based on the temperature. For example, saprophytes into these are low temperatures, less than 25%. I mean, sorry, less than 20 degrees centigrade. And mesophils, these are where you have 25 and 30 something. And by the time they are reaching up to from 55 degrees centigrade and above, that is a thermophile because they are thermophilic. You know, I made mention in DLC, I made mention of something which is called PCR, and I said about a thermophile. There are some microorganisms that at very high temperature, that is when they will begin to perform well. That is the environment they like so much. That is because they are thermophilic. So thermophilic organism or microorganism will not perform well at low temperature. So, and those ones that are classified that they are saprophyte, they will not perform when the temperature is getting higher. So you should know that. And these are what we call the optimal temperature. Then in terms of pH and osmolarity, so you can also classify them into allophiles and osmophile. Osmo means that they have that ability to just maneuver, to assist, to diversify. So in terms of their alkalinity or their acidity. Then oxygen, the presence of oxygen. Oxygen, when you say they are obligate aerobies, when you come across the word obligate, obligate means it is obligatory, it is important. So that means they are obligatory aerobies. They cannot live any other life apart from being aerobic. And there are some that are also classified as obligate anaerobes. They cannot live without or with oxygen. A little content of oxygen to them is going to be poisonous, it's going to kill them. So, and when they are obligatory, they are obligate aerobies. That means they cannot exist without oxygen at all. So, you should be able to know from the word obligate. Obligate means it is compulsory, it is mandatory. They cannot live without it. So, and when we now say facultative actually means that they can they can exist in both. So when you say they are facultative aerobes, that means they can exist as in the presence of oxygen or in the absence of oxygen. So if they are facultative aerobes, fine. One, as long as they are getting oxygen, they will continue to exist. And whenever they don't even have oxygen any longer, does not mean that it's going to kill them. So they will just switch into the new, they will adapt easily. So that is what is called facultative. And when we say micro aerophiles, micro from the word small, the smallest quantity, aero of oxygen. So they exist with smallest. Once they have the smallest quantity of oxygen, life continues with them. Lives continue with them. And those that are aerotolerant, that means they can tolerate oxygen. They are not affected by the presence of oxygen. Okay, then we will also look at these later on, or it might be in this course, or it might not be in this course. But what we have been saying so far is a media types and the aseptic technique. As a microbiologist, you're supposed to know how can you grow, your microorganism in the laboratory. For example, it's just like you that want to domesticate, you want to have dogs. You want to have your pets. So when you now have your pets, if you want your pets to be able to perform well, you must know the food that they give them. So the same thing with microorganisms. Don't forget that microorganisms exist outside the laboratory. Although some of them are present in the laboratory, but those that are present in the laboratory, they exist in their natural environment. But you want to study them, you want to have them, you want to see the way they look like. So the best thing for you is to be able to define, you be able to know the food that they eat. And the food that they eat is what is called the media, the food that they feed on in the laboratory. Because that means you must, for example, now, if you want to rear a lion, there are so many things that they are carnivorous. When they are carnivorous, that means they feed on they are flesh eaters. So if you want to bring lion into your house and you want to just be you must look for a way that you will be feeding them with exactly almost everything that they feed on in the jungle. So the same thing with a microorganism. So microorganism, you are bringing them into the laboratory. You must know their food. You must know their best food. You must know their favorites. And their favorites must be that you must supply them with a source of carbon, which could be in form of carbohydrates, source of carbon, some Okay, sorry, it was a result of technical issues, and I want to believe that that will be resolved later. Okay, that will be resolved later, and sorry about that. Sorry about that. That means the next lecture we will take, the next lecture we are going to, I'm going to be a bit, I'm going to be a bit patient that I will go and make sure that I also start from where you can actually link up, where you can link up. So um, don't forget, who is this Yusuf Rashid? Who's this Yusuf Rashid? I don't know what you mean by 250 1462. I only need your name. I don't know any other thing. So please, I don't want to see anything like a number here. What I want to see is just your name and your name only, nothing again. So yeah, I can see some. I can see Adekunle, see Taiwo, Akin, okay, Taiwo Shekinat, Aisha Adekunle, Oluwaseun, Segun Adebayo. I can see you. I can see you. Favor Oshibajo, I can see you. Babalola Israel, I can see you. Oyetunji Kehinde, I can see you. Adebayo Deborah Ayomide, I can see you. Aisha Adekunle, can see you again. Oluwasegun Adebayo, can see you. Yeah. Deborah Ayomide, yes. Olamilekan Ayo, I can see you. Okay. There are Babalola Israel, can see you. Can see you. Kafayat Adio, Folashade, I can see you. Okay, I can see you. Marvelous Oladipupo, I can see you. So, yeah, Rashid Yusuf, yeah, I can see. Yes, I can see. Saprophyte cannot grow in higher temperature, truly, while thermophile can grow in lower temperature. That's good. That's good. Okay. Um, that's good. That's that's good. Okay. Yeah. Yeah. I quite agree that at a point that is Adelayo Elizabeth that you lost the connection. So sorry, we will make up in the next lecture. Sorry about that. Uh, okay, I can see you have technical issues. It is very possible here. Okay. So the class is ending the class. I'm ending the class. I've ended the class already. The only thing is that I need to take time to be able to respond to some of the comments I have in the comment section. So don't forget, we will have to meet again, and probably we'll meet again on Friday. On Friday, by the grace of God, and I'll be able to make a review, general review of this, and so that I'll be able to understand where you have actually lost here. So once again, thank you for your time. See you some other time. The Yeah, the conversation can still continue in the WhatsApp group that is on the platform. And that platform, I might also decide to give you one or two things, because I actually want to know whether you are actually getting what you are actually getting along in the understanding the class. So I want to test that. So let's meet in the WhatsApp for further discussion. So once again, thank you. God bless you.